I'm Daniel Levine, and this is the Bio Report. GLP-1 agonists have been a breakthrough for the treatment of obesity, a global problem with serious health risk. While the benefits of these therapies are seen as outweighing the risk, there have been some concerns about the loss of muscle mass, particularly in older people using these drugs. iBio is developing next-generation obesity therapies that may be able to be used in combination with GLP-1 agonists or by themselves. Its lead experimental therapy is a myostatin inhibitor, part of a class of therapies that have been of interest to treat muscle wasting. We spoke to Martin Brenner, CEO and Chief Scientific Officer of iBio, about obesity, the limits of GLP-1 agonists, and the potential for myostatin inhibitors alone or in combination with GLP-1 agonists. Martin, thanks for joining us.
Thanks for having me today, Dan.
We're going to talk about obesity, iBio, and its AI platform technology that uses computational biology and 3D modeling for antibody discovery. Let's start with obesity. How big a need does this represent today?
Okay, so let's start with some numbers then. In 2022, the WHO estimated that there were 890 million obese people worldwide. That is 1 in 8 people in the world living with obesity. So you can imagine that that is a significant problem that has grown over the last decades. The health risks caused by overweight and obesity are increasingly well documented and understood. They include cardiovascular diseases, diabetes, cancer, neurological disorders, chronic respiratory diseases, and others as well. The financial impact on the healthcare system is enormous. A study in a journal called PLOS1 from 2021 estimated that the annual obesity-related medical care costs in the U.S. to be nearly $173 billion.
We've seen a major breakthrough with the GLP-1 agonists. How much of an advance did they represent, and what are the limitations of these therapies?
So GLP-1 receptor agonists have been indeed a major breakthrough in the treatment of obesity. Before the GLP-1s, or more precisely before the incretin class of drugs, patients had to undergo gastric bypass surgery, which is invasive and permanent, to reach the 20 to 25% weight loss that now can be reached with GLP-1s. With the GLP-1s now, we finally have a pharmacological alternative to this surgical procedure. But in general, I do see 2 limitations in the use of GLP-1 receptor agonists. So let me start with complex diseases and single drugs. There's not a single complex disease that we can actually treat successfully with a single medicine. And obesity is quite a complex disease. So first, there is an adverse effect profile of the GLP-1s, mainly focused on gastrointestinal adverse effects, including nausea, sometimes vomiting, reflux, constipation, diarrhea, and along with other more rare risks such as pancreatitis. But they have been well documented. In fact, the insurance company Blue Cross Blue Shield conducted an analysis over a 10-year period and found that almost 60% of people taking GLP-1s stopped using the drug after 3 months, and nearly a third stopped taking the drug within a month, which is actually before you see meaningful effects on body weight. The second limitation I see is that GLP-1s can cause a loss in muscle mass and bone density. And as we age, we lose about 5% muscle mass in a decade, starting around the age of 30, and this decline actually accelerates after the age of 60. Now, clinical trials have shown that patients can lose up to 10% of their muscle mass within 16 months only when treated with GLP-1s, which equates to roughly 20 years of muscle loss due to aging.
Before we talk about what iBio is working on, I wanted to make sure certain concepts were understood. What is myostatin?
So myostatin is a member of the so-called TGF-beta superfamily, or transforming growth factor superfamily. This group of molecules plays a really crucial role in regulating a lot of biological processes, from embryonic development to tissue homeostasis, which the muscle comes into play, and also immune responses. So myostatin itself is one of the two major negative regulators of muscle mass in humans. The other one is activin A, but both belong into the TGF-beta superfamily. For us, myostatin is so incredibly interesting because it is what we call a genetically validated target. What this means is that there are people in different ethnic groups that have a genetic loss of function of myostatin, and those individuals are lean, they have significantly increased muscle mass, and they're very strong. Beyond that, they don't seem to have any adverse health effects, which makes blocking myostatin such an exciting mechanism.
Well, there's been a lot of interest in myostatin inhibitors to treat muscle wasting diseases, but as you mentioned, there is this turn to looking at it as potential for obesity treatments. This is iBio's lead experimental therapy for obesity. It inhibits myostatin. Can you explain what it is and how it works?
Yes, absolutely. So before we actually jump into the anti-obesity treatments with myostatin, we have to consider the long history of anti-myostatin molecules. It all started with myostatin inhibitors being used for muscle wasting in patients with cancer. And more recently, they have been used in neuromuscular diseases like spinal muscular atrophy. And some companies are now trying to repurpose those molecules for an obesity indication. Now, repurposing molecules for an obese population, as you can imagine, comes with some significant challenges. First, rare disease patients often have no alternative treatment, so the acceptable route of administration is usually more invasive. Most of these molecules are actually administered intravenously, whereas an obese population actually has access to incretins, which are being dosed subcutaneously, self-administered, which is a lot less invasive and more convenient, can be done at home by the patients themselves. Second, the patient population of these rare diseases is, at least in the case of spinal muscular atrophy, about 10,000 to 25,000 patients in the United States. And you don't have to produce a lot of antibody to actually serve this patient population. And your manufacturing process can actually be quite inefficient. But if you want to treat, say, 10% of an obese population, which is almost 100 million people, you have to produce tremendous amounts of drug substance, and you have to be extremely efficient in your manufacturing process. Both of these challenges can't be easily fixed after the fact, and quite often the route of administration and the manufacturing efficiency have to be designed into a molecule very early in the discovery process. So when we set out last April, we were the first company to really design a myostatin antibody specifically designed for a large population. So the molecule is called iBio 600. It is a long-acting anti-myostatin molecule designed for infrequent subcutaneous dosing. And our early manufacturing data also show that iBio 600 is very well behaved, indicating that we can develop a highly efficient manufacturing process and produce iBio 600 in large quantities. We are very excited about the preclinical data from iBio 600. In a recent study in obese and aged non-human primates, iBio 600 has not only shown a significant extended half-life of 41 to 52 days, which is about 3 to 4 times higher than the typical half-life of a human antibody in monkeys. And with this data, we utilized two mathematical models to predict the human half-life of iBio 600. So it is estimated between 57 and 130 days. As seen with other antibodies, this extended half-life could enable dosing as infrequently as every 3 months or every 6 months. We are also the first to demonstrate that 8 weeks after a single dose of iBio 600, monkeys equivalent to 60 to 70-year-old humans showed a muscle mass increase of up to 270 grams or 0.6 pounds. This exceeds the gains observed with other anti-myostatin inhibitors, which required multiple weekly doses in younger monkeys. The use case for anti-myostatin drugs, and specifically for iBio 600, which is differentiated due to its profile in obesity, is kind of threefold. First, we can prevent the loss of muscle mass in patients on GLP-1 drugs. Second, we can treat patients after they stop taking GLP-1 drugs. GLP-1 drugs do have adverse effects that actually lead patients to rotate off them quite frequently. And then last but not least, because we believe we have a differentiated profile for iBio 600, we can also use anti-myostatin as a pure single anti-obesity play as well. It has actually been shown in preclinical models that myostatin blockade alone can not only increase muscle but also reduce fat mass. But that has still to be seen in humans.
What's the development path forward?
So the development path is really that we're planning to do the toxicology studies at the middle of the year, this year in the summertime, which then gets us to the first patient dosed, likely in the second quarter of 2026. We will start very traditionally with a phase 1 study that assesses the safety first and foremost of iBio 600 and its half-life in humans, but we also intend to see early signs of efficacy in this phase 1 study that will then set us up for phase 2 study in obese patients.
It may surprise some people that iBio has long been a public company. It was a 2008 spinout of Integrated Biopharma as a public company. It was long focused on using its platform technology for producing biopharmaceuticals in plants, but in 2022, the company acquired Rubric Therapeutics. How did that deal come about?
Yeah, you're absolutely right. So iBio is first and foremost an incredible turnaround story from a manufacturing organization to a modern AI-enabled antibody discovery company. From 2008 to 2022, as you said, iBio was a CDMO, a plant-based expression with a plant-based expression system producing antibodies and vaccines. And in 2021, when I joined as the chief scientific officer, my main goal was to basically build a biotechnology arm here in San Diego that would directly feed programs into this manufacturing facility in Texas. So Rubrik came into the picture. I was aware of Rubrik's technology from my previous company, and I always felt the technology was very intriguing. We first signed a license agreement to develop one of our oncology assets, as well as a discovery collaboration. And we were very impressed with the capabilities of the Rubrik team and also their technology platform. If you will, Rubrik was a pioneer in AI-enabled antibody discovery, and they were several years ahead of the field. But of course, with a deteriorating financial environment, external environment, the company got into trouble. And in September 2022, we had the tremendous opportunity to buy substantially all of Rubrik's assets. And more importantly, we were able to relocate their key scientists to our R&D center here in San Diego. The acquisition enabled iBio to really then pivot from if you will, a CDMO into a modern biotech.
So, what was the thinking from a strategic point of view? How did you see this remaking what you were doing at iBio?
So really, the asset acquisition brought a very, very sophisticated early machine learning discovery platform, an amazing team, and some preclinical assets to iBio. If you will, that kickstarted our efforts to really become an AI-enabled antibody discovery company. And the goal really was to drive our preclinical pipeline into our preclinical programs into the clinic, while creating also a business around partnerships for targets that are really hard to drug, which our platform enables us to do.
So, this is not just a matter of increasing the speed and efficiency of discovery, but you think this will get you to molecules that you would not have been able to discover through more traditional approaches?
Absolutely. So the the AI hype we have seen in recent years has really led to big misunderstandings what AI can deliver in drug discovery and development. Think about it this way: it takes more than 10,000 steps to make a medicine. At iBio, we enable 3 of these steps with AI. Does this make us an AI company and our molecules AI drugs? No, it doesn't. But it enables us to make medicines we couldn't make before we had access to, say, generative AI models that we have access to now. But our technology stack can deliver real outcomes and just hypotheticals, right? So a lot of platform companies have platforms that have actually not delivered differentiated molecules. So we can talk about speed first. So we have developed Bio 600 from idea to a development candidate in 7 months. That's a process that usually takes more than 2 years. And if you do employ more traditional discovery processes. But we can also talk about creating antibodies against targets that nobody else was able to create. So take, for example, our actin E antibody. This is a very exciting genetically validated target that is thought to produce a fat-specific weight loss. So not like the incretins that also lose muscle mass. This is coming purely from fat. At least that's the idea. So you can imagine that the industry was buzzing around this target. And despite the immense interest, nobody was able to create an antibody against this target until we came around. It took us about 3 months to come to the first functional antibody in that space. So we can actually enable targets that we couldn't do before. We actually applied that AI technology.
Give us a sense of the discovery process. How does the platform work, and at what point do you move something into a wet lab?
Mhm. So our strength is really the combination of machine learning tools with the wet lab. So we have multilingual scientists, if you will. They're not only doing the wet lab part, they're also doing the machine learning AI part. So we usually start with our epitope steering engine. Think of this as almost like Google Maps for antibodies, right? It allows us to steer our antibodies against the correct address on a drug target. Drug targets are massive proteins. There are many, many regions that are non-productive. They have no biological function apart from keeping the protein in its structure. But if you have antibodies against these regions, they have no biological function. So we can actually steer our antibodies specifically against the regions that do have a biological function. And that allows us, if you will, to remove a haystack and just go straight to the needles. In the best case scenario, this enables us to find molecules that nobody else can. In the worst case scenario, if I want to speak about worst case, it just makes us a lot faster. Others can catch up, but it makes us faster. Once we have identified an antibody, we usually need to optimize it. And antibody optimization usually takes 4 to 8 months if you basically use more traditional processes. We have broken this time down to 3 weeks. And the way we've done this is by very cleverly combining machine learning tools with wet biology. So what we have is basically a so-called mammalian display technology that allows us to put antibodies on top of a cell. The cell is later being used for manufacturing. So we know every antibody that comes off of this library of antibodies, we already know we can make it in manufacturing. We have also the opportunity to optimize these antibodies in multiple characteristics all at the same time. We call this a single shot multidimensional optimization. The way this works is we're shooting these cells through a cell sorting machine, and at the same time, we're looking for antibodies that have high expression levels, so are made in large quantities in the cell, bind very tightly to the target we want them to bind to, not bind to any other target. And then also we introduce what is called species cross-reactivity. That's really important for the safety assessment of an antibody. Our main safety species in antibody discovery are non-human primates. So we always build into our antibodies this cross-reactivity, not only targeting the human protein, but also basically recognizing the protein in non-human primates, which then lets us assess the safety of the molecule a lot better. Now, the downside of this mammalian display technology is that we can only fit about 1 million molecules in there. So you have one input molecule, and then you build basically cousins, closely related neighbors of this molecule. And if you only have 1 million to basically select from, the chances of finding ones that are better in multiple parameters is smaller than using a traditional technology where you have billions of molecules. And this is how we combine machine learning. We take these larger libraries and basically compress into much, much smaller libraries that then will fit into our mammalian display. And this is what generative AI is really good for. And so this combination of both technologies, wet lab and the machine learning, really allows us to be differentiated faster and also pick antibodies out that later on will be much, much better to manufacture.
We talked about the Rubric deal, but another deal I wanted to ask you about was the licensing agreement with Astral Bio. You licensed iBio 600 from Astral Bio, but
Mhm.
this was discovered using your own AI platform. Can you walk us through the relationship between iBio and Astral Bio?
Absolutely. So Astral Bio approached us last year with an idea for a multi-target discovery collaboration for cardiometabolic diseases and obesity. After evaluating multiple platforms, Astral Bio concluded that our platform and our team was best suited to, discover those best-in-class and first-in-class antibodies. The collaboration really consists of Astral Bio identifying 4 targets. iBio then would use its technology stack to identify and rapidly advance these assets from inception all the way to in vitro proof of concept, so in cells. And then Astral Bio can actually continue the preclinical development and advance these candidates. But at the same time, iBio has the exclusive option to in-license 3 of these 4 candidates. And then we would receive the exclusive global rights to develop, manufacture, and commercialize those candidates. As you mentioned, the first molecule in-licensed was iBio 600, and we have the right to in-license 2 more. So we have full control over the antibody discovery, which is made in-house. So we know the antibody very well and its characteristics. And at the same time, the licensing fee of $750,000, which we paid in common stock, is actually a tremendous deal for iBio, specifically because we know of all of the characteristics of this molecule.
In addition to the $750,000 payment, there was a potential milestones of $28 million. It seems like a modest deal by most biotech standards. How should investors read this?
Yeah, so I would call this a tremendous upside deal. As you know, usually these deals come with much larger clinical milestones and royalties. Obviously, for potential licensing clinical assets, taking royalties early on is not a good idea. Obviously, Astral Bio has the rights to payments, but vice versa, if we decide not to license a program, we have the rights to the same terms. And we consider this actually low payment if we license these molecules in. And it all started because the Astral Bio collaboration really started with Astral Bio helping us to recapitalize the company last year.
You talked about the recent preclinical results for iBio 600, but you also had some results on your activin E antibody. Can you explain what that is and what you've seen so far?
Mhm. So activin E is a really challenging molecule. It is proteins have to fold to have a function, and this molecule does not fold easily when you make it outside of the body. So we used our technology platform to work around these issues and found the first functioning activin E antibody. We first tested it on human fat cells, which is the proposed target tissue for activin E. And we have seen that it actually reduces or basically completely blocks the signaling of activin E, our antibody, in these human adipocytes. This is important because using human cells or primary human cells gets us very close from a so-called translational perspective to dosing humans. So this is not mouse. This is not a different species. These are true human cells, and that give us, a lot more confidence in it, in the molecule working. Next, we moved this molecule forward into a mouse study. And what we've seen is the antibody alone causes a relatively mild body weight loss of about 8 to 9% at maximum. But it all comes basically just from fat loss. These animals lost a tremendous amount of fat and no loss in muscle mass. Of course, we've also treated and used this molecule on top of a GLP-1. And what we've seen was a dramatic weight loss after only 2 weeks of almost 35%. So imagine a human being weighing 100 kilograms, so a regular, slightly obese person losing basically more than 30% body weight within weeks. And what was more important than this is all of that weight loss, the 35%, more or less came out of fat. There was only a very, very minor component of muscle loss. This actually replicates very nicely the data that competitors have shown, specifically companies that use knockdown of the gene that produces activin E in the liver. And now we have basically confirmed that finding. But obviously, antibodies have a lot more flexibility to be developed to medicines than we see with liver-specific knockdown.
You joined iBio in 2021 as the chief scientific officer. You became CEO as well in 2023. Your background is that you've worked for several big pharma companies, AstraZeneca, Eli Lilly, Pfizer, and Merck. But what was the attraction here? What did you see as the opportunity?
hmm So when I joined as the CSO, what attracted me first was that I truly had a blank canvas. I could start with a blank canvas. So building a top scientific team and a research site that was really tailored to what we needed to do, that has been a big part of me joining iBio. And then, of course, when I was offered the CEO role in 2023, I was aware of the nearly unsurmountable challenges at the time, the deteriorating external environment, the debt we had on our balance sheet, manufacturing facility, which was located in a tier 3 hub that nobody showed interest in buying, and then also a very, very high retail shareholder base. But I also saw the enormous talent and passion of our team. I saw the technology platform that could transform how we create medicines, and the conviction that if we carefully craft a disease area strategy, that would lead us to overcome all of those obstacles.
iBio seems like a lot of small biotechs today that may have promising candidates or platform technologies, but are sitting with market caps well below $100 million. Does being a public company help or hinder your ability to raise money and execute a plan today?
I would consider the external environment of the last maybe 5 years being beneficial for being a public company. It has actually enabled us to raise money and execute our goals. But I think what is really important is you need to set yourself apart. You mentioned this. There's many companies out there that are doing similar things. So how do you differentiate from all of the other competitors? I believe what sets us apart from other companies is that we successfully merged a platform technology with disease area expertise. There are a lot of platform companies that have no differentiated drugs in development. They're brilliant platform companies, but they're currently facing questions about the value of their platforms. If you cannot produce a differentiated molecule, what is your platform worth? And then on the other hand, there are asset companies with molecules that have maybe progressed into the clinical development, and they're struggling to rebuild or build and execute a solid preclinical strategy. I think this is what really sets us apart. We have a clearly defined obesity strategy. We are extremely efficient with our resources, and we're fast, and we can actually drug undruggable targets. And that in combination truly sets us apart and actually gets us into meetings with investors that companies of our size usually would not get.
What's the discussion with investors like today?
So first of all, we have strong relationships with our larger and current institutional investors over the last year. We have completed a capital raise last March with them, and that has given us a lot of credibility. We have also been able to prove that we can execute on the plans we have proposed. But when we speak to new investors about iBio today, what they really like is kind of this underdog turnaround story. They like that we have a platform that has actually delivered, and not just for the platform's sake. And we have also shown that we can be extremely fast, capital efficient, and that we can create these differentiated molecules in a reasonable amount of time. And I think that combination gets us into these meetings that we're currently having.
And you mentioned the capital raise. How far will existing cash take you, and what's the plan for raising additional capital?
Mhm. So our current cash runway is July, August this year, and we're currently in discussions of a potential fundraising round, which we anticipate to allow us to advance iBio 600 into clinical development at a minimum.
Martin Brenner, CEO and CSO of iBio. Martin, thanks so much for your time today.
Thank you so much, Daniel. Really appreciate it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.